This lecture is the Science Communication lecture, specifically focused on science writing for the Bio 282 Microbiology Lab, Spring 2026, here at USM. So, we've just done a few experiments. Now you've made an amazing discovery. Now what do? Typically, we want to tell somebody about it, right? So we've got to communicate the results. How do we do that? So there's several ways to communicate your results. Scientists will communicate their results through scientific writing. That's either in journal articles or news articles. So peer-reviewed journal articles can either be primary sources or secondary sources, and news articles are always secondary sources. We're going to talk about those types a little bit later because we're focusing on this today. And then there are two other ways to communicate your results that are common in science that we will talk about later this semester. There are poster presentations, which are done at conferences or symposium sessions. This is typically preliminary data, student data, short-term research, things like new methods that you've developed or things that you're looking for feedback on. This is more of an informal setting where you've got more one-on-one conversation. Then you have oral presentations. These are thought of as more prestigious. These are your traditional PowerPoint assisted presentation where you're standing in front of an audience for 15 to 20 minutes giving a talk. The layout is similar to a journal article in content. However, you're typically more focused on the intro, where you're giving the background or why you did your research, and your results of what you found. So, scientific writing, the parts of a paper. We're going to start with the abstract. The first thing you would read is the abstract. Sometimes it's the only thing you read. So this is the elevator pitch. This is the one paragraph summary of everything that happened through your entire experiment. So if this is your thesis research or years of research put together for one paper, it's still only one paragraph. It's very short. This is a very succinct, concise summary of everything you did, why you did it, how you did it, and what you found. Then comes your introduction section. This is the why. This is the background info. What the reader needs to know to understand the topic you're about to get into. Why is this research important? Why did you bother doing this? It would end with your hypothesis and a lead into your results in the microbiology field. Methods. Methods is sometimes called methods and materials or materials and methods. For us in APA format, we're just going to call it methods. So this is the how, specifically your recipe, your protocols, your procedures that you followed. So you want to be very, very detailed here. Somebody should be able to read your method section and be able to replicate your experiments. This is not a numbered section. This is still written in paragraph form, but any products or kits that you used, you should be able to say, we used Promega's DNA kit purchased from XYZ. We should be able to say, we incubated it for 24 hours at X number of degrees. You should be able to say exactly what you did so that somebody could follow it. Results. This section is the what. Your data and observations only. So this is what you found. You're not including any analysis to it. You're just going to walk us through step by step of what the important information was that you found from this research. The discussion is really where you dig into those results. What did they mean? Why did they come out the way they did? Errors, methods, did something go wrong? Was it a reagent? Was it user error? Was it what's going on there? And why is it important? How does this relate back to the real world or back to your hypothesis or back to the question? How is this important? And finally, a references section. We're going to dig into it. Obviously, plagiarism is really bad. You will understand by the end of this, science has a different definition of plagiarism than the rest of the world. In English, you probably learned how to quote things and understand that that's acceptable. It is not acceptable in scientific writing. So we will talk about that in the references. So these are all of the parts of your paper, abstract through references, and we will get to write one of each of these this semester. Digging a little deeper. So your abstract section. So this is, again, short, concise. Really think of it as a challenge to summarize your entire experiments in as few words as possible. This section should be able to stand on its own. You shouldn't need anything else to be able to get a full picture of what happened. So it should start with your background and research question. It should have the results and why it matters. And this little picture sort of breaks it out into colors. It should have context, content, and conclusion. So you should have the one question you're answering. Here we do, what we found, this is how it matters. Your introduction section. So remember, when you're writing your abstract, there is an introduction section and a method section coming after. So it's okay to leave out a little bit. You can really make it short and to the point because you're going to expand in the very next section. So your introduction, it provides that context for the reader. What background info do you need to know to really understand this experiment? Start very, very broadly. Then focus more specifically to your research question. Pretend you're explaining this to first day you. Remember when I asked, what are microbes? What's the very basic definition? And you guys all kind of looked at me like I was insane. And we had to break it down word by word. You need to start broad. Explain it like you're an honors student coming into college. You don't have microbiology yet, but you're perfectly intelligent enough to understand a college level course, a high level high schooler sort of thing. So this is where the bulk of your literature research goes. Most introduction centered sections are just sentence, citation, sentence, citation, sentence, citation. You don't need to add citations for well-known facts, like E. coli is gram negative. However, you do need citations for not well-known facts, like this strain of E. coli has an inserted plasmid that produces a glow protein or something. Citation by this author. So be very specific after every sentence where that fact came from. Methods. What are the exact instructions you followed to get to this result? Somebody else should be able to read these, replicate your experiment exactly. So this is typically written in subsections. So this little notepad here explains the different heading levels in APA format. So all of your section headers should be level 1 headings in APA format. So your abstract, introduction, methods, results, and discussion and references should all be words that are centered, bold, just like this methods. They should all look like that. They're all going to be the same size font as everything else, but they're going to be centered and bold. That's a level 1 heading. For your subsection header, you can group the next group. I've grouped this a couple of times to give you examples. So a level 2 heading is fermentation ability tests. So I'm grouping anything that shows the microbe's ability to ferment things is underneath this heading. So fermentation ability tests is a level 2 heading, so it's going to be left aligned and it's going to be bold. And then I've grouped the tests again by another subsection header underneath that. So to tell us exactly what each test is underneath that group. So those are level 3 headings. So those are going to be left aligned, bold, and italic. They continue on, and there's more resources. You can look up what a level 4 heading is, and you can keep going down and down and further on if you'd like. So I've grouped level 1, level 2, level 3 underneath my level 2 heading. And then I've gone back and I have a level 2 heading for my next group of types of tests, and then a level 3 heading for the test that's under this type of test. And then I've got my instructions here because this is our methods and we're telling them how you do the test. There is the biggest common thing that gets messed up in the methods section. So again, be very specific. You're telling them how to do the test and how to interpret it. Not what your results are. Your results do not go in the methods section, but you would tell them how to interpret what the results would be for these tests. Results. This is your what. So this is only a statement of the data. So all of your figures and tables would go here. That helps you see the data. Your figures and tables don't go anywhere else. So they shouldn't be in your methods, and they shouldn't be in your discussion or your introduction. Occasionally you might have a map like in your introduction to show where your sample or where a region is or something like that. But it's pretty rare. All of your figures and tables traditionally go in your results section. Be very clear that no interpretations are happening in the results. So you're not explaining why a test didn't work or why the results came out the way they did or why you did multiple trials. This slide is really nice to tell why you put the pictures in the results. So basically you've got your data and it gets collected and it's just a big mishmash in your lab notebook. You maybe started sorting it out in your lab notebook and grouping it together into different places. You maybe even put it in tables in your lab notebook so that it's sort of arranged nicely. Then you put it in a table in Excel so that it's presented visually and you're like, okay, so this starts to make sense. Then you explain it with a story with your discussion. So these are the presenting it in a figure or a table. This is your lab notebook part. This is the explaining it in the discussion. So this is the sort of visual of why it's important to put your data and visuals in your results area, and then your discussion to really explain them later. All right, discussion. This is the explain. Explain what the results actually mean. You're going to make sure that you discuss any errors, any difficulties you encountered, compare your results to published results. Do they follow what's already been found, or is it completely different? What would you do differently, or what would you do next as a result of this experiment? If you had all the time and money in the world, what's next? What are you curious about now? Sort of thing. References. So don't plagiarize. For science, putting it in quotes isn't enough. I never want to see quotes in any of your papers. If you put quotes, that is considered plagiarism in science writing. It is not acceptable to put quotations in a paper in science writing. So you must use in-text citations, and you must paraphrase what you have found and write it in your own words with an in-text citation at the end or beginning of the sentence. You can say something like, this author, parentheses, date, said that blah, 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 or found that da, 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 da. Here are two different types of sources. These are talking about peer-reviewed journal articles, right? So peer-reviewed journal articles. Are scholarly sources. They have gone through extra rigorous testing by experts and have been reviewed and been analyzed and been corrected. Primary sources are reporting experiments, studies, new research. They're giving us new data and giving us new information. The key information here to figuring out if they're a primary source, they typically will have a methods and a results section, because they have to tell you how they got the new data and then tell you what that new information is in the results. So these are your clues that it's probably a primary source. Secondary sources are always summarizing other primary sources. So they may give you new information that you don't know, but they're summarizing somebody else's new information. They're maybe bringing it all together in one summary document. Sometimes they will have the word review or summary in the title. Don't get confused, because sometimes meta-analysis or meta-review, meta-summary will be in the title, and those are actually primary sources. What you want to look for is really descriptive headings. So if this doesn't have your typical introduction, methods, results, discussion, if it says something like the purpose of the cell membrane in protein transport, that's not a typical abstract introduction results heading. That's really descriptive. That's probably a secondary source. Then we have textbooks and news articles. So these can still be reputable or scholarly sources. So like a textbook can be written by a doctor, can be written by some other researcher. So these can still be reputable, still be scholarly, but they're not primary. These are still secondary sources. So they're still summarizing other primary sources. If the news article or textbook, specifically if a news article is talking about scientific findings, you have to go find the original journal article, the original peer-reviewed article the findings were reported in, and cite that. You can't just cite the news article. You will not typically cite a news article in any scientific writing. It's not an acceptable citation in science writing. Textbooks are an acceptable citation in science writing, but news articles are not. If you're looking to meet a required number of primary sources, a textbook is not a primary source. This is a secondary source. Is this reputable? Yes. News articles, not a source I want to see on your science writing. Okay? All right, so different types of methods of studies. These are all different ways that you might set up your experimental design. So this might be your traditional experiment. So you might have a control, you have an independent and dependent variables, and you're testing a hypothesis. You set it up in a very controlled environment, you only change the one thing, and you see what happens. You might have an observational study where you record data as you observe it in its natural environment with no outright test happening. You might have a case study, which is interpreting an individual historical case to get a better understanding of a topic. So this typically is in medicine when there's a weird case that happens, something that hasn't happened before, hasn't been observed, and how it was handled in the hospital at the time. Then you have correlation studies. So it measures relationships between different variables. So this happens a lot. This is when happens in people where there's too many things going on, or it's unethical to do a traditional experiment on them. So we measure as many variables as we can, and we find the relationships between those variables and do a bunch of math to figure out which variables have the most effect. Then you have the ones I was sort of warning you about, the systematic review or meta-analysis. So it has the word review in the title, but this is not a secondary source. This is still a primary source. So they're going to review current research, but they're going to analyze that data. So what they're doing is they're going to use their data collection, is their source is going to be all of the data that's out there. All of the current research, they're going to use that as their current data collection pool they're going to pull from. So they're going to have a methods section that explains how they chose which sources to study, like which things made their study and which things didn't. Then they're going to do some sort of statistical analysis, some analyzation of that data, and they're going to report out results and come up with new conclusions about the current state of data. So they are still providing new research on this information, even though they did not physically conduct what would seem like new research. They are actually doing it electronically. There's many, many more types of studies that can be done. These are just some pretty obvious ones that you're going to probably run into. This one is very common and gets people tripped up all the time. But just remember, primary sources typically have a methods and results section. Secondary sources typically have section headers that are more descriptive about the topic. Sometimes you will notice that the journal itself says reviews. Sometimes you will notice that the title of the journal article will say a review, but don't get messed up about the systematic reviews or meta-analysis. Here's the most common science writing issues that I find in this class. Formal tone. Formal tone is the biggest issue I find. People write in a very informal, sort of poetic language. They write as if it's a creative writing English paper. Science writing tends to be very boring. And that is hard for some people to adjust to. It is very cut and dry. It doesn't tend to have a lot of flowery sort of language to it. It does not talk in the first person. No I's, no U's. If you are struggling with this, see me, see a tutor. The Writing Center is very helpful. This is something that I totally accept the use of AI for. Write your paper. If you feel like your formal tone isn't there, ask AI to edit your paper for formal tone. The other one is formatting of bacterial names, scientific names. When you write it out, a lot of people will abbreviate, and abbreviations are okay, but make sure that you write it out the first time, the full genus and species, the first time you mention it, and then you can abbreviate. So this nice little graphic is cute when your mother calls you by your full name, Escherichia coli, and it goes shit, and it's got a little E. coli next to it. So we've probably heard it called E. coli before. So you have to spell the whole thing out the first time you write it out, and then you can substitute and just shorten it to E. coli. So when you abbreviate, you can only abbreviate the genus. You cannot shorten the species at all. So when we have other species like B. subtilis, Bacillus subtilis, you cannot shorten subtilis. So I do not want to see anybody abbreviate Bacillus subtilis as B sub or something like that. The other thing is that when you write it in words, genus and species get italicized, but only the genus gets capitalized. The species should all be lowercase. So that's my only gripe with this comic, is that coli should all be lowercase. Only the E should be capitalized. And same thing here. This should all be lowercase, but this whole font is capitalized. Table and figure formatting. People mess this up all the time. There's lots of resources for this. I think I get very clear on it, and there's lots of examples. Again, don't use quotes, don't use quotes, don't use quotes. You must paraphrase this in your own words. Practice this by reading something, looking away from the screen, and actually use pen and paper and write it down. Or just flip screens. So navigate away from the window you're in and write up what you remember and flip back to make sure you don't have the same sentence written down, but that you've still got the essence of it held in your words. And then people mess up the primary versus secondary research articles all the time. So primary is presenting new information. Secondary is summarizing other people's new information. So primary, you typically need at least 2 primary sources. So you need new research on most of our papers. Secondary sources, you can have as many as you want. But you need at least 2 primary on most of our papers. Tools. There are lots of tools. So many things to make your life easier. Zotero. This is a references and citation management program. I suggest you download it. It's fabulous. It integrates with Word. It integrates with Google Docs. You basically just hit add citation, and then you type either the author's name or the name of the article that you're looking for, and you hit insert citation. It asks what style you're using. You select APA 7th edition, and it inserts the citation in the paper in text correctly. And then at the end of the paper, you just say add bibliography, and it will add your references section correctly in the correct alphabetical order, all cited properly for you. So you don't have to memorize how to write an APA format. You just need to be able to glance at it and double check. Zotero is wonderful. You don't need... I learned how to write references back in my freshman year of high school, and they made me buy index cards and a one of those plastic card holders with the recipe indexes, and we had to write each of them down by hand and organize them in a little like Rolodex style. I'm not making you guys do that. That's ridiculous. We have tools now. You don't have to know how to do it. Use your tools. Our library website has a thing where you can just click and copy the citation in the correct style. Use your tools. On our library website, there is a button where you can click Interlibrary Loan. It's also known as ILLiad. You will have to create an account. It is separate from your USM account. I highly suggest having an interlibrary loan account. If there is an article that you can't get access to, so it's just not something that we have, you can request access. So you put in all of the information you have about it into ILLiad and you send it off. And typically within 24 hours, our librarians will have a digital copy for you waiting in your email. And they're pretty amazing. Between interlibrary loan and some other work, they were able to find me a PhD thesis of somebody from 1936 for my thesis work. It took them a little bit longer. It took them about a week, but they were able to find it. It came as a physical copy, as a book, but all the other ones came as a digital copy. They can find it in other languages. So if you can read a language other than English, it will even ask you, do you want this in other languages? So if they can't get it in English and they can get it in French or German or whatever else, they might be able to get it for you. Interlibrary loan is awesome. So if you find something and it looks like it's going to be perfect for your paper, but you can't read the whole paper, throw it into interlibrary loan. You might be able to have it by tomorrow, and it might be what you needed. Google Scholar is such an easy first step. It does a really good job of finding new sources and just having access to them. I always start there. And then I go to our library page to see if it's something that I just didn't, like Google Scholar didn't have access to, but our library does. Because Google Scholar is just easier to navigate. There's a resources tab on Brightspace, the ones that I curated, the one that school curated, the Writing Center, there's tutoring help. For sure, the Writing Center. Go schedule a Writing Center to help. Like if you're stuck and you're just staring at a blank screen, they'll help you start writing. If you want help editing it, they'll help. All of the things. They are very, very helpful. There are USM accessible AI tools right from your Launchpad. At the very bottom, there's like a triangle in rainbow colors. That's Gemini. That's Google's version of ChatGPT. It does not train its model on anything you put in there. Go ahead and use Gemini to edit your papers, things like that, for outlines, for helping you format. Notebook LM is really great for helping you research things, for pulling in research, and for making PowerPoints and quizzes and study guides and stuff like that. Though that's right next to it on the Launchpad. So those are things that USM has for you. Other commonly used AI tools I've seen used in this class and cited for me have been Grammarly and ChatGPT. I do not use those. I have no experience in them, but they have been used pretty frequently for this class. But if you need help with either of the two USM ones, I'm happy to help you. Those are my thoughts. Those are my things from science writing. You guys are going to be writing an abstract after we finish up your couple of experiments with microbes in the environment. You can kind of pretend that these two experiments you did were like two trials, so to speak. You don't have to talk about it as far as a time limit goes. You don't need to say that they were done over 2 weeks or anything like that. You can just say 2 experiments were performed, or 2 trials or something like that to that extent. Try to challenge yourself to get as few words as possible when you're writing your abstract. My thesis was limited to 250 words, and I spent 2 years on it doing research. So your 2-week research should certainly fit in under 250 words. I challenge you to as few as possible. If you have any other questions, please feel free to email me. I hope you have a lovely weekend.